Welcome Bio 105 students. This is the DNA puzzle, which you probably recognize from class. This will be a demonstration of DNA structure, DNA replication, when it happens and why it happens, and DNA's function. Okay, so let's take a look at the pieces. If you can't read these clearly, it's not a problem. We've taken pictures of everything, and that should be at the beginning of this presentation, so you might want to rewind later on. Okay, this stuff right here, you may remember, is deoxyribose. D, like deoxygenated blood, meant without. Oxy, of course, means oxygen. So deoxyribose is a sugar that is missing one oxygen. In a minute, I'll compare it to the ribose so you can see what I'm talking about. Another thing you should take note of is that the OSE ending indicates a sugar. And this I just happened to mention was a pentose sugar. That means it has 5 sides like the pentagon. The next pieces we have are the bases. The bases come in 2 different types, purines and pyrimidines. It's not important for our discussion that you know which are which. More importantly, that you need to know that they only fit together in certain ways. You'll notice that A can only go with T, and C can only join with G. Okay, remember also, we can't see well on these puzzles. These are 3-dimensional molecules. There's depth and width and length to them. And that's important in how they fit together. Okay, you'll notice that the A with the T, the G with the C. Okay, next we have phosphates. The phosphates are going to help hold all those steps of DNA, the rungs of the ladder that we just made, out of bases. These are what are going to help to string it together along with the sugar. That's DNA's components. Now we move into some of the other puzzle pieces which will come in handy later on. You may remember these from the puzzle. I'd like you to just disregard these, okay? We didn't use them. Are they important in real life? Yes. Again, we're trying to get a concept. Here's the pentose I mentioned before. This sugar, which also has 5 sides, is called ribose. If you compare deoxyribose and ribose side by side, you'll notice that the dark DNA sugar is missing this oxygen here. That's why this one is deoxyribose and this one is just ribose. This goes into DNA, ribose goes into RNA. So you can see where the molecules also get their name from. These pieces here were like the adapters. These were transfer RNAs. And each transfer RNA has its own unique 3D shape. These will help bring amino acids in the correct order to the DNA. This was a substitution that was made when you were making messenger RNA. The messenger RNA needs to move quickly, and you use uracil because it provides less molecular resistance and can move a little faster. Second to the last, we have some of the amino acids that are in our body. The amino acids actually come in 2 different types. There's essential amino acids, and those are the ones that you must get from your diet. So that's why a well-varied diet is so important. And then there's non-essential amino acids. That means your body can make them. Basically, whatever their source, the amino acids are what will link together to ultimately form a protein. Okay, here, this thing you may remember we didn't use it. This represents a ribosome. But you know what? It was so large and cumbersome, I switched to this. Okay, so here we have the 2 subunits of a ribosome. The 2 subunits of the ribosome will join together and clamp onto the messenger RNA. Okay, the next thing we're going to do is build DNA. So what we'll do is we'll take these pieces, which I've prearranged. These pieces would actually be floating around in the nucleus for a cell that has a nucleus, or if you're a cell like a bacterium that doesn't have a nucleus, you'd be floating around really close to the chromosome. These pieces come from your diet, and your body makes a lot of them too. All right, so what's the first thing to do? I guess in my opinion, let us start with joining some bases together. Okay, so you notice that we have an A, the straight letter, with T, a straight letter. That was my little learning tool. Each one of these also needs a deoxyribose. So there we have the beginning of our first step in the DNA ladder. Okay, what I'm going to do is to continue to build the DNA, and then we'll pause and we'll take a look at what I built. Okay, as I'm finishing up our puzzle, you'll notice DNA. I ran into a few technical difficulties, the same things you ran into in class. For example, the pieces don't fit together snugly all the time because they came from different batches of puzzles. The other thing is I don't have enough pieces to be as ambitious as I thought. We may in fact have to use our imagination, like we did in class. Okay, so let's take a look at our DNA molecule. You'll see it's basically made of a bunch of repeating units that look like that. That little thing is called a nucleotide. When you start linking nucleotides together, that's how you get a nucleic acid. So a nucleotide would be considered the subunit of DNA, and we'll find out RNA also. You'll notice it's made of a base, a sugar, and a phosphate. Okay, so keeping that in mind, let's take a look. First thing that comes to mind is what made me decide to put these in this order. Well, what happens is that you could do it randomly, but you probably would just get garbage. I happen to know for a fact what these units actually mean. Okay, so we keep that in mind. The next thing is what holds the two sides of the ladder together. These dotted lines that you may remember from the puzzle refer to hydrogen bonding. Hydrogen bonds are nice because they're strong enough to hold them together, but still they're breakable. For when we need to open up the DNA for replication or open it up for copying a gene. Okay, the next thing is you'll notice there's two sides to the DNA puzzle. DNA will ultimately take a perfect mathematical spiral that's called a double helix. The DNA pieces, each rung of the DNA ladder can't sit right on top of each other as indicated in this puzzle. They kind of have like, they kind of repel each other a little bit and push each other to the side, so you end up getting this spiral molecule. Okay, next thing is something we talked about in one of my classes, but not both classes, was how much of this pattern, which makes us who we are, is unique to individual humans. The statistic comes down to something about 99.9% of the DNA from one human to another is identical. It's only that 0.1% that differs between people. Identical twins typically have identical DNA. Okay, another thing is you might say, okay, well, how much do we have in common with a chimpanzee? The answer comes out to something like 98.8% identical to a chimp. Don't get upset. Don't go asking about the family tree. What happens here is that if you look at a chimp, they have two eyes, a nose in the middle of their face, they have a mouth, obviously. Their internal organs are very similar. They have the opposable thumb. They have fingerprints. Yeah, clearly we're going to have a lot in common. Then you could go down to something like a dog, where the number varies depending on your source, but an average number would be somewhere around 60% in common with a dog. Think about it. It's a vertebrate. It's a mammal. We're vertebrates. We're mammals. We share a lot of the same characteristics. Now let's go way down to something like a mushroom. Yep, we have DNA in common with a mushroom. They're not plants. They actually have to digest their food like we do, so that they would have some enzymes in them that are similar to ours. Let's pause for a second. If you think about it, fungi are not plants. They digest their food using some of the same enzymes we do. The enzymes are protein. And ultimately, what is the function of protein? The function of protein is to hold the code, the information, to make the proteins that run metabolism. This 3D pattern, and I know it's kind of 2D-ish here, but this 3D pattern is the information to make the proteins that run metabolism. Okay, let me think what else. I think we're good there. The next topic we need to discuss is DNA replication. DNA replication occurs just before the cell divides. As we discussed in class, if you have one cell, that means there's one set of DNA. When this cell gets big enough, it has to divide and make two. So the question is what do we do with the DNA? If we were to split the DNA, that wouldn't be very good. That means that each new daughter cell would only have half a set of DNA. You can't run a cell with half the information. If we use my cookbook analogy, if DNA is the cookbook, if we split it like this means one cell would get all the even pages of the cookbook, the other cell would get all the odd pages of the cookbook, and you wouldn't be able to do anything. Same thing with DNA. So what has to happen, when the cell gets large enough, and it either has to divide or rupture, what you have to do, what happens is there's chemical signals sent to the DNA. An enzyme will start cutting the hydrogen bonds, unzipping the DNA molecule. And then just as quickly as the DNA is being split, another the complement base to each of the base pairs starts to form. And basically what you're going to do by the time you're through with this, and I'm not going to build the whole thing, I just want you to get the idea, is by the time you're through with this, you're going to have 2 strands of DNA. I'm out of light blue. Oh, no, here's one more. I was going to say we're going to have to use our imagination again. Okay, so, and we'll pop in the phosphates. So by the time you're through with this, by the way, these stacks are like chips at like the poker table or something. Okay, you're going to have 2 strands, and I'm just going to add a few more pieces so you get the gist of it. And here, and whoopsie, and here, and here. There we go. That's enough for me. Hopefully that's enough for you. If we look at these side by side now, if I fill in all the sugars, okay, and all the sugars on this side, you would have 2 identical strands of DNA. That's awesome because that means you have a full set of instructions for one daughter cell and a full set of instructions for the other daughter cell. The whole thing is regulated, mediated by those proteins called enzymes. Okay, the last section that we're going to cover is DNA's function. There are 2 fancy words that I used. One was transcription, one was translation. You're not responsible for those words, but I would like you to know that the process is divided into 2 different parts. As you look around the table, you'll notice that I brought in the cast of characters that was involved in DNA's function. Let me remind you what they were. Over here, I brought in a ribosome. The ribosome was 2 big chunks of protein that need to clamp down ultimately on the messenger molecule that we make, which is called mRNA. Okay. The next thing is, and I labeled it rRNA to stand for ribosomal RNA. Another piece that we have was this guy right here. Okay, there's actually 2 parts to this. This molecule right here was a transfer molecule, and this was an amino acid. What you'll notice is that the shape of the transfer molecule is unique. When I compare this to another transfer molecule for another amino acid, you'll notice that they're clearly different in shape. What that means is that this shape can only carry this amino acid, and this shape can only carry this amino acid. That's kind of nice because that means that we can't screw up too much when we're putting our proteins in order. And I'll tell you what happens, or what can happen if you should mess up the order of those amino acids. Another player in functioning DNA was when we make our copy, the mRNA, which is a copy of a gene. What happens is that this substitution, the uracil, goes in place of thymine, okay, or T, and if you see, they actually do have the same shape. And as I mentioned, scientists believe this has less molecular resistance. You'll notice we're using the other sugar. In this case, this was ribose, and this is the one that has the oxygen on it. Otherwise, the phosphates are the same. Let's just take a look at our DNA a sec. One thing that can happen, and it'll mess up everything, is called a mutation. What happens is a mutation changes the sequence of bases in the amino acid, in the DNA. So what can cause those? Ultraviolet light, that's how people get skin cancer, X-rays, chemicals in food and cigarette smoke, other kinds of radiation, etc. And what can happen is a base can just get popped out by the strength of those mutagens, they're called. Your body's really good. It has enzymes that go around and they kind of can feel. And they go, whoa, 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 whoa. Okay, we have this shape here. It's up to me to put the shape back. And it can repair your DNA, and it does it all the time. What can happen is a couple of different things. One thing is that you could lose the whole rung of the ladder. Your body doesn't have any way to compensate for this, so what it typically does is it just moves things up. And then what you like cut these out and just move this up, but that's bad. If the letters are in a very particular pattern to tell me what protein to make, if I take this out, it's like taking a letter out of a sentence and trying to read it. It's not going to work. Okay, so that's that. The next thing is this is a big, big section of DNA. You have to use your imagination. These can be hundreds of thousands of bases long. It is composed of small sections. Each section of DNA, in this case, it happens to be this chunk right here. How do I know that? Because I made it. This chunk right here is a gene. If you take gene after gene after gene and put them all in sequence and attach them to each other, that's your DNA strand. If you take your DNA strand and coil it super, super tightly, I use the example of the telephone cord getting twisted and it makes knots and then the knots fold on the knots. That is what forms a chromosome. So sections of DNA called genes join together back to back to back to make your DNA strand. The DNA strand coils up and now it's called a chromosome. Okay, so let's take a look again, remind ourselves, what is the information that DNA carries? It's this 3D message right here. Okay, what we want to do is, we went to the gym, we need to make some muscle protein. And this right here, our gene, is the gene to rebuild our muscle protein. If you think about it, when you lift weights, you rip your muscles open and now you have these gaps that you have to fill in. You fill them in with protein. That's what makes your muscles bigger. Where do you get the protein? Your body makes it based on the recipe from DNA. Okay, so here's our DNA for a muscle protein. We'll call this myosin, because that's an important muscle protein. Genes don't just open up all the time, or don't get copied all the time. They only open up when they have a need. Clearly, some genes are going to be needed almost all the time for your entire life, but some will only open up, like when you work out and need extra protein when you need them. Okay? When a gene is actually being copied, it's called being turned on or being expressed. When the DNA is closed, like it is here, the gene is turned off, or it's not being expressed. Okay, so we have a need, we have the gene, what's going to happen? Okay, what will happen is an enzyme will start opening up the DNA, and then very, very quickly, you're going to make a copy of the DNA template. Why make a copy? Sounds like a lab question. Why make a copy? Your DNA is too valuable to be cutting and pasting and moving around. You want to keep this safe. So you need to make a 3D molecular copy. All right, so what your body does is it starts very, very quickly building a copy, making the substitute, oops, I'm not going to start there, I'm going to start here actually, guys, making the substitutions that we talked about. Okay, so here I am, quickly, I am making some substitutions. I'm using my uracil in place of T, because normally A goes with T. I'm using my ribose instead of my deoxyribose. I am, however, using my same old phosphates that don't seem to fit very well. Okay. You might ask yourself, how did she know to use this side of the DNA and not this side? Well, I got to tell you the truth. I'm kind of making this whole thing up as I go. So I was just playing. I was just guessing. In real life, scientists know which side of the DNA, for the most part, has to be copied. Okay, guys, so what I just did is I made my molecular copy of the DNA of a particular gene, in this case, myosin. Good. Okay, next thing is, you may see I keep going like this. I'll explain these in just a second. Next thing is, this molecule now is called messenger RNA. What we have just done by copying the gene is that first process I mentioned, transcription. Okay, so what happens, and remember, this is happening in like microseconds. It only feels like hours, right? So I'm going to move my messenger RNA out of the nucleus if the cell has a nucleus, or away from the chromosome in bacteria, and out into the cytoplasm. The cytoplasm is the jelly-like filler where many, many chemical reactions take place. The other thing that has to happen is the DNA has to zip back up again. Okay, so I unzipped it to read the gene, and now I'm zipping it back up because I'm done copying the gene. If you actually did go and lift weights, this gene would be copied multiple times throughout your muscles. Okay, but we're not going to do that. What I'll do here is I'm simply going to use my imagination and get rid of that, and now I'm out in the cytoplasm. Okay, so now we are using our imagination and we're in the cytoplasm. Okay, that's where these characters come into the game. Okay, so we have our transfer molecule with its specific shape and carrying its appropriate amino acid. And I mentioned where you get the amino acids before, so they're coming from your diet, they're floating in the cytoplasm of your cells. Okay, the ribosomes are also in there. You get your first set of ribosomes from mom. They're in the egg. Thanks, mom. Okay, so that's that. One thing that I'm not doing here is the messenger molecule needs a lot of preparation. So there's going to be chunks in it that might not make any sense. There's going to be, you have to attach something at the beginning so you know when to start, and then you add something at the end so you know when to stop, etc. Okay, so we're just cutting right to the fillet right here. Okay, so in your cytoplasm, what has to happen is the ribosome has to clamp down on your messenger molecule. It's clamped. Okay, so 3 bases at a time, in case you were wondering, why is she doing this? It's called a triplet or a triplet codon or a codon. It takes 3 letters to code for 1 amino acid. So what happens is, with the ribosome in place, these molecules, these transfer molecules, can now come over. That's not the right one. But how about, look at that. So the message from DNA, as it's being read or translated, that's the second process, says bring me this amino acid and bring it to me first. So I'm going to attach that to my ribosome. The transfer molecule will go back into the cytoplasm if it's still in good shape, and it will pick up another of its amino acid. Then the ribosome scoots along to the next 3 bases, the next codon. And what do these say? Let's see, do they say bring me this? They do not. Do they say bring me this? They do. So the message that originated with DNA says bring me this, bring it to me first, bring me this, bring it to me second. Okay, and we're going to use our imagination. There we go. Now the ribosome scoots off. The last 3 letters or the last codon says bring me this amino acid and bring it to me third. So we add that to our chain. And this could go on for hundreds of thousands of triplets, giving you a very, very long polypeptide or protein chain. When you reach the end, there's another couple of letters that say stop. When that happens, the protein breaks off, the ribosome can disconnect, and your messenger RNA can be reread. Sometimes you can even have several ribosomes reading it, one right after another or almost simultaneously going along it. If your body says, okay, I have enough of that myosin, that muscle protein, it can start using enzymes to take the mRNA apart, and these pieces can be reused and recycled. In the meantime, the amino acid chain, your protein, can go to where it's needed and do its job. One other thing I mentioned was if these get in the wrong order because of a mutation, that's really not that common. If you actually have a mutation, typically you're not going to get any protein at all. It's going to stop wherever the mutation is. That means your protein stops, and it probably won't be functional. Another thing that can happen is in the case of something like hemoglobin, if one of those substitutions that I mentioned early on when I was talking about mutations, if that occurs, you end up getting your amino acid chain, but it's incorrect. What has to happen is many of these chains have to fold on each other. So in the case of your red blood cells, you need to have your hemoglobin fold perfectly so it can carry oxygen well. Folks who have sickle cell anemia have 1 letter in their DNA messed up, which means you have one letter in your messenger RNA messed up. It means that when you're putting your amino acids together, one is wrong. That's enough to change the folding enough so that the surface of your red blood cells that hold the hemoglobin can't hold your oxygen well. That's what sickle cell anemia is. Okay, that concludes my presentation. Check the review questions and check the animations I gave you and check the cheat sheet that I gave you. Signing off.